Hi everyone, welcome back to part 2 of chapter 3 on biomacromolecules. And we ended chapter 1 by talking about having gone through carbohydrates and a little bit about the lipids and stopped at the phospholipids because there are 3 specific subtypes of lipids. There were fats, phospholipids, and steroids. We've gone through and done the first 2. Now we're going to get to the last subtype of lipids called steroids. So their shape is very unique compared to the other 2 types of lipids. They usually have at least 4 linked carbon rings with a short tail sticking out at the end. Because they are made up of a lot of carbons and hydrogens as well, they are also hydrophobic and hence are insoluble in water. So one common example of steroid is cholesterol, which is the most common steroid synthesized by our liver. It serves as a precursor to a lot of other types of steroid hormones, such as testosterone and estradiol. Estradiol is just another name for estrogen, vitamin D, and bile salts as well. You see that there are 4 linked carbon rings right there, and then you have some sort of a short tail at the end of the molecule. So those things I've been underlining as I'm talking through them are sort of the key things or characteristics you can look for in the image to help you remember which specific type of macromolecule it is. And hence these are specific molecules that can be made and are also considered as part of the steroid group of lipids. Testosterone, your male sex hormone, major male sex hormone. Estradiol or estrogen, your major female sex organ, sex hormone. And then your vitamin D right here, which is essential molecule for bone growth that you will, again, something we'll talk about when we get to your anatomy and physiology if you're going down that route. Okay? All right. So that's all we have about lipids. We will now move on to talk about our third major type of biomacromolecules, and probably is the most diverse type of biological macromolecules compared to the other 3. We have proteins. Okay, proteins. So proteins is something that has a huge range of functions. For example, when we talked about carbohydrate, lipids, and nucleic acids, we can narrow down to usually 1 or a few functions. But for proteins, the possibility is endless. Our cells, organs, body use proteins for multiple number of different functions, such as structural purposes. For example, when we use it to build things or support tissues, like collagen fibers underneath our skin, all those collagen fibers are actually made up of proteins. For contractile purposes, which is movement per se, those proteins are specifically myosin and actin in our muscle tissues that allows us to move. Okay, if we don't have those proteins, we can't move. For protective purposes, we have antibodies that are manufactured by our immune system to keep us healthy and fight off infections, and we'll talk a little bit about that, sort of a quick glimpse and gloss of that when we get to plasma membrane. So there's also proteins for enzymatic reactions that allows for production of many different things in our body, like ATP in the process of cellular respiration that we will talk about later in chapter 7. Okay, there are just etc., many different types of proteins that has different functions in the body. The possibilities are just endless. Now, even though the functions and structures are, they can vary quite a bit in proteins, what monomers constitute the protein polymer are monomers called amino acids. Okay, just as what we have. Monomers for carbohydrates are your monosaccharides, your monomers for lipids, depending what it is. For example, fats, we had glycerol and fatty acid chains. So for proteins, the monomers for proteins are amino acids. So proteins are basically linear long chains of different combinations of amino acid sequence that will interact with each other with those chemical bonds we talked about from the last chapter to form a three-dimensional protein structure. Okay, so there are just this linear chain. If we just stretch out all proteins, they're really just little balls of amino acids linked together by, again, covalent bonds, and there will be specific names for these. And then they will fold on top of each other into a three-dimensional shape like this that will make it functional. Okay, something I have already talked about in the last chapter, or mentioned quickly in the last chapter. And so within each amino acid molecule, and within each amino acid molecule there are 5 different parts to it. Okay, just like how we talked about that there are different parts in a phospholipid, there are different 5 major parts in an amino acid structure. You have to know the basic structure of the amino acid. Okay, so you have your central carbon. You don't have to know alpha carbon, just know it as your central carbon. You always have a hydrogen attached to the central carbon, and then you have what's called the amino group. Okay, amino group, which is basically NH2. Okay, that's attached to one side of the carbon, and then the other side is what we call a carboxyl group. Okay, this whole thing right here, which can be easily written as COOH, like that. Okay, and then the last thing that attaches to the carbon, remember the bond tendency for carbon we learned from chapter 2 is 4 bonds. Okay, so one is attached to hydrogen already, we talked about. One is attached to amino group, one is attached to carboxyl group, and the last thing that the carbon is attached to is a side chain that we call the R group. Okay, because there are 20 different possible amino acids that can take the place of this side chain group of an amino acid. Okay, so between the 20 different amino acids, all of them share this basic structure. Okay, they all have an amino group, they all have this hydrogen right here, they all have a central carbon, and they all have a carboxyl group. What differs between them is this R side chain group right here. Okay, so that's where the different amino acids will differ, and how the side chains differ between the 20 amino acid is their chemical structure or nature, which can be either nonpolar or polar. So some groups are nonpolar, which is what this group is showing you here. All of these here are considered as nonpolar R groups. Okay, nonpolar R groups, and you can see there's no charge on them, and they're usually made up of just carbons and hydrogens, and with here in this particular one with a sulfur group. There can be charge groups where it is positive charge or negative charge. Okay, so this group right here is positive charge, where you can see a positive charge somewhere along the side chain of the amino acid, or negative charge, where you can see a negative charge somewhere along the side chain. Or this particular group here is a polar group, meaning that there is an even distribution of charges due to the electronegativity we talked about, and these are the polar groups here. Okay, polar side chains, all of these here. So again, notice that all of them share that same central carbon, amino group, carboxyl group, and hydrogen. All of them have that. What differs is the blue shaded regions in each of these. Okay, and the last group of side chain here is what we call aromatic R groups. Aromatic, that you know something you'll learn in chemistry. Aromatic usually means they have a ring of some sort. Okay, carbon ring of either hexagon or pentagon, depending the case. Okay, so these are the 3 amino acid that has a ring structure. So the interaction between these side chains, that all those different chemical interactions we talked about in the last chapter, is what forms that basic 3-dimensional structure that I will talk about in detail in a bit. So hence, it is the sequence and number of amino acids that will determine the protein shape, size, and essential, most of all, or most important of all, is function. Okay, and remember I said the very important thing to remember about protein is structure is to function. If you have the right structure, you're going to have the right function. Okay, if you don't have the right structure, you're going to lose that function. Okay, because a lot of times how proteins work is based on their 3-dimensional structure. So what we mean by these interaction, I kind of talked about it before, but I'll re-emphasize it here. For example, if you have a sequence of amino acids in a linear sequence of amino acids, if one amino acid right here, for example, let's say have a side chain that is positive charge, and another amino acid right here, let's say have a negative charge side chain. Well, we talked about how opposite charges attract each other, so they will tend to come together like this because one is positive charge, one is negative charge. They will form an interaction, ionic interaction with each other. And when these 2 amino acids comes together, it will also bring in the rest of their amino acid sequence, creating a bend like this in the 3-dimensional shape of the protein. So that's just a very simple illustration of what we mean by the sequence of amino acid is what will determine the final 3-dimensional structure of the protein due to the interaction they can form between different amino acids. Okay? All right. Okay. So we talked about, how covalent bonds are formed between 2 sugar molecules are called glycosidic bonds. And then the bonds in the triglyceride molecules are called ester bonds. And hence the bonds between amino acids that link them together, covalent bonds, of course, in a protein molecule are called peptide bonds. Okay, easy way to remember that is it starts with the same letter as proteins. Okay, so peptide bonds in proteins, glycosidic bonds in, for example, glycogen. Okay, so that's how you can easily remember the names of these bonds for different macromolecules. When multiple amino acids come together, they will form a peptide chain that we call a polypeptide chain. Okay, remember we said how monomers come together in forms of polymers, and we talked about carbohydrates are formed by monosaccharides forming polysaccharides. So for proteins, amino acids can come together to form polymer called polypeptide chain. Okay. All right, so same thing across the board. Whenever you have formation of a bond, it's called dehydration synthesis reaction, and you are removing a molecule of water from the 2 monomers. So here's an example of amino acid 1 here, and then another amino acid, and the carboxyl group supplies the hydroxyl group right there, OH, and then the amino group supplies the H. So this is what's going to come out as a byproduct as a water molecule. Okay, water molecule. And then what will form the bond between them is this one right here, between the carbon of the carboxyl group and the nitrogen of the amino group of the other amino acid. Okay, and that particular covalent bond right there is called the peptide bond. And this carboxyl group can then go on to react with another incoming amino acid, and that amino acid would have an amino group as well. And then the same process repeats, where this OH group and the H group from the next amino acid will form a molecule of molecule of water in dehydration synthesis, form another peptide bond, and continue on and on and on until you have a whole linkage of polypeptide chain making up of many different amino acids, okay, in linear sequence. Now, don't forget that formation of bonds is achieved by dehydration synthesis, okay? And because a protein structure is determined by the side chains of amino acids, there are 4 levels of protein structure that these interactions can create. Primary structure is basically the amino acid sequence in linear format within the polypeptide chain. So that's what I mean. They kind of look like beads in a necklace or something, and each one of those beads represents one amino acid. Okay, so that's why there's all these little 3-letter abbreviation. These 3-letter abbreviation stands for that particular amino acid, and we have 20 of them to choose from, and different proteins will be comprised of different amino acids. So, for example, insulin is a protein that has 2 polypeptide chains, chain A and chain B. They're linked by a disulfide bond right here and right here and right here as well. Okay, that will give its further 3-dimensional shape. But if we lay them all out, this is what we call by primary structure, the linear sequence of the protein itself. Okay, so sequence of the amino acid is what the primary structure means, and it is very important for its function, because what sequence it has drive the function, drives the 3-dimensional structure of the protein, which is very important for its function. Okay, all right. Remember, I've emphasized from last chapter that what's critical for a protein to function properly is its structure. You must have the correct 3-dimensional shape in order to do its job. Like what I mentioned about Legos. Remember, I used that analogy. If you miss a few pieces of Legos in your, Lego model, the final product may not work as it should. And I've done this before with my son, where he forgot one little tiny piece in his car Lego model, and something just didn't work properly. When he rolled the car, something's supposed to be moving and nothing moved, and all that was missing was just that one little piece in that one particular location in order to give you that function of a movable piece. Okay, so just like proteins is analogous to that. If it's not in the shape it's supposed to be because it missed that one important chemical interaction, you lose its function. Okay, and in some cases, changes in just one amino acid in sequence of a protein is enough to completely disrupt the whole normal structure of the protein, and hence its normal function. And sometimes that can ultimately result in what we call a genetic disease. A classic example of this type of change is in the disease called sickle cell anemia. In normal red blood cells, we have very important protein for carrying oxygen molecules in our red blood cells called hemoglobin. Okay, so inside our red blood cells, within there are lots of tiny little proteins called hemoglobin. Okay, and they kind of looks like this. squiggly lines, three-dimensional structure of these polypeptide chains that resides within the red blood cell itself. And this particular protein is supposed to have a three-dimensional shape that allows it to bind to oxygen efficiently, so that we can transfer and transport oxygen all over our body and distribute it to every organ system. So here is a part of the protein hemoglobin, normal amino acid sequence right here. This is normal hemoglobin molecule amino acid sequence, and with valine being the first one, histidine, leucine, threonine, proline, and then you have glutamate right there. So when the sequence of the hemoglobin is correct, it will fold up in such a way that will give the red blood cell a very nice circular structure. Okay, that's why we see red blood cells at that circular structure, just like this right here. And then they will be able to deliver oxygen efficiently. Okay, but with the disease sickle cell anemia, however, what happens is that this glutamate right here in the 6th position is switched with a completely different amino acid called valine. Okay, so this usually happens due to what we call a DNA mutation. And for how mutation occurs and why DNA mutation results in a different protein structure is a process we will talk about in the latter part of the semester when we talk about genes and proteins, which is also where we will talk about cancer as well. Okay, which is stems from mutations. Now, here in this particular case of sickle cell anemia, there is only one amino acid change in this particular location. Of course, different sickle cell anemia cases may have different locations of mutation, but this is just one example. And that one change in one amino acid completely end up disrupting an important chemical interaction, and it changes the shape of hemoglobin such that you end up with a very flat and elongated red blood cell instead of a circular cell. Okay, so that's why you see this is a microscopic view of the red blood cell. In a normal red blood cell, they're supposed to be circular like this, but when you have sickle cell anemia cells, sickle red blood cell mixed in with it, you see these elongated shape cells in there as well that look very irregular. Okay. And, with these elongated cell, they're called sickle cell anemia because their red blood cell kind of shaped like a sickle. A sickle kind of looks like this with a little handle down there for harvesting wheat and something, or rice fields, things like that. So that's what we call it, sickle cell anemia, because a red blood cell end up looking like a little sickle. And this dramatic change in shape results from just one amino acid difference in a protein that is made up of about 574 amino acids total. Okay, so one hemoglobin molecule, just like this right here, okay, in this structure, this is one hemoglobin molecule and is made up of 574 amino acids approximately. One difference in amino acid can completely disrupt this interaction between these proteins right here, polypeptide chains within the hemoglobin molecule, that gives you odd and irregular shaped red blood cell like this and makes oxygen delivery inefficient. Okay, so it's kind of hard to imagine, right? But how one amino acid difference is all it takes for you to end up with something that you have to deal with for the rest of your life as a genetic disease. But that is how important the sequence of protein, of amino acid I talked about. Okay, it is very critical that our DNA sequence is in normal sequence of different nucleotides that we'll talk about when we get to genes, which determines which amino acids gets incorporated, which determines their final three-dimensional structure of your protein. We will come back again to this idea and the amazing way our cells have to minimize this type of amino acid change or chances of mutation in a protein structure later in the semester. Okay, but this is a glimpse of how a mutation can detrimentally affect an organism or like in us. Okay. And hence, the ultimate effect in the normal function of the hemoglobin is its efficiency in carrying oxygen. When the red blood cell looks like this in this circular format, it is very efficient in carrying oxygen. But if it looks like this elongated sickle cell shape here, it is a lot less efficient in carrying oxygen. And that's why individuals with the disease usually can't participate in heavy sports, because they can't supply as much oxygen to their tissues and organs as a healthy individual would. The other important effect that can happen is red blood cells are shaped in this circular way, because it allows them to squeeze through teeny tiny blood vessels called capillaries. Some capillaries in our body can be as small as the diameter of a hair strand. Okay, so you can imagine how small that is. So the way red blood cells are shaped allows for that smooth blood flow as they are moving through all of our blood vessels regardless of their diameter. But for those with sickle cells, what happens is because of their abnormal three-dimensional structure like this, they tend to clump against each other. They will stack on top of each other and potentially block blood flow like this by forming a big blood clot. And we all know that blockages of blood flow can be a serious matter depending where the blockage is located in the body. And hence they have to take special medication. Individuals with this sickle cell anemia disease, they would have to take the special medication to minimize clot formation. Okay, whereas in normal individuals with normal red blood cells, their blood flow is very normal through their blood vessels, whereas in sickle cell they can form clumps like that. So all those different functional effects in sickle cell, and it's all due to one single amino acid difference in the overall sequence. So remember, for proteins, it is always what structure it has, then that's the specific function it has. And the structure is accurately determined by the primary structure of proteins, which are just a linear sequence in the order of the 20 different possible amino acids. Okay? All right. So the secondary structure of proteins involves two different basic types of folding in three-dimensional shape. One is called alpha helix, the other is called beta pleated sheets. And both of them are formed by bonds called hydrogen bonds between amino acids of the protein. So they're held together by hydrogen bonds. If they're double helix like this, okay, or it's not double helix, but alpha helix, you have hydrogen bond here, hydrogen bond here, hydrogen bond here, for example, to connect the different coils of the helix together. If you have a beta pleated sheet within the protein's secondary structure, then the hydrogens are formed between the pleated sheets like that here, and those dots, okay, the dots between the hydrogen and the oxygen. Okay, so then the tertiary structure is formed by what we talked about earlier, those multiple variety of different types of chemical interactions or reactions that can occur between side chains of different amino acids within the protein molecule. So we talked about how we can form hydrogen bonds. Disulfide linkage is a type of covalent bond that can occur between two sulfur groups. And the hydrophobic interaction, remember we talked about van der Waals interaction. An ionic bond that can form between two opposite charge, anion and cation. that we talked about in chapter 2. So some of these interactions we talked about are those examples of hydrophobic interactions, such as van der Waals. We talked about ionic bonding between cations, which is positive charge, and anions, which are negative charge. Hydrogen bonding between hydrogens and oxygen, or hydrogens and nitrogen sometimes. And disulfide linkages are example of covalent bonding between two sulfur groups, sulfur atoms. Okay. And the last level of protein structure is called quaternary structure, which is when multiple polypeptide chains come together to form a final protein. So keyword there is multiple polypeptide chains, or what we call subunits. So some proteins are not just made up of one single amino acid polypeptide chain, but multiple of them can come together to form a functional final protein. For example here, collagen fiber actually, those protein that sits underneath your skin I talked about earlier for structural support is actually made up of many polypeptide chains together. Here's one particular collagen subunit, but 3 of them can form what's called triplex helix, okay, that form your collagen fibril, and many of these can come together to form your whole one single collagen fiber, or, really, if we look at it accurately speaking, is collagen bundle of many, many, many different polypeptide chain that makes up the collagen fiber. And it is same for hemoglobin. Hemoglobin is not just made up of one polypeptide chain, but is actually 4 polypeptide chain. 1, 2, 3, and 4 here. Okay, so 4 polypeptide chains come together, or we can call them subunits as well. 4 subunits come together to form your final structure of hemoglobin molecule, which is why one hemoglobin molecule can bind 4 different molecules of oxygen. This little purple coin looking thing here is where oxygen binds. And one of these is one polypeptide chain being able to bind one oxygen molecule. And one hemoglobin molecule is what we can have 4 different oxygen molecules binding to it. Okay? All right. So now along the line, so we talked about how important a protein's 3-dimensional shape is for its function. Not only can that structure be compromised by changes in the amino acid sequence that we talked about, for example, with sickle cell anemia due to DNA mutations, but also sometimes different environmental conditions can also change a protein's 3-dimensional shape and structure. Now, the process where the protein loses this 3-dimensional shape due to disruption of those important chemical interaction we talked about is called denaturation, or the protein has denatured, okay? And the environment factors that can change or affect a protein's 3-dimensional structure include temperature, pH, exposure to different chemicals, whatever that can disrupt those chemical interactions we talked about, disrupt the ionic bond, disrupt the hydrogen bonds, etc. And so when you have a functional protein, it should have all these little coils and 3-dimensional bends, etc. within the protein structure itself, okay? That's why the hemoglobin molecule looks like globular shape like this. But a denatured protein loses all those 3-dimensional folds and bends and result in maybe a single chain of your primary structure of the protein, okay? And when you lose its shape, it loses its function, okay? And something we can actually see denaturation of is if we think about an egg, okay? Before the egg white, if we look at egg white is mainly made up of proteins. When you first crack the egg white of the egg is actually clear. It's see-through because there are active proteins within the egg white in their globular shape with all those chemical interaction that gives them this particular 3-dimensional structure. But when you heat the egg up on the pan, heat is changing the temperature of the environment of the protein, which denatures or disrupts those interactions that are formed within the molecule itself. Hence, the protein unfolds into its linear shape, or what we call the protein has become denatured and, in a sense, inactive. And that is why the egg white, once the pan is heating up and changes that shape, all that egg white becomes white is because the change or denaturation of the protein that changes the reflection of light that now is no longer clear and see-through, but it is now white in color. Okay? And sometimes some protein is, once it becomes denatured, there's no going back. Once the egg is cooked in that white color, there's no way we can turn that back into opaque color or clear color. Okay, so some denaturation of a protein is permanent, while other protein can actually, after denaturation, they can be formed right back into its original active form. For example, in warm milk. Okay, once you cool it down, it stays within their correct configuration. Okay. All right, so we'll talk more about denaturation when we get to the topic of enzymes in metabolism chapter, or within our lab on enzymes. So here's a summary of the 4 levels of protein structure again to recap. We have your primary structure of the protein, which is just a sequence of amino acids. Secondary structure has to do with formation of alpha helix or beta pleated sheets. And then you have tertiary structure, which are then the 3-dimensional folding pattern of a protein due to those side chain interaction. Okay, so notice that this is the side chain interaction that forms a tertiary protein structure. And then quaternary structure has to do with protein that consists of more than one amino acid chain. So 2 or more polypeptide chains coming together to form the final structure of a protein that we talked about, for example, in hemoglobin. Okay, so this is again the example following your primary structure, sequence of amino acids, secondary structure, either alpha helix or beta pleated sheets, and then you have tertiary structure, which is a side chain interaction that gives a 3-dimensional shape, and quaternary structure, different subunits coming together to interact with each other. Okay. And finally, we have our last group of biomacromolecules for this chapter on nucleic acids. Nucleic acids is actually the most important macromolecules for continuity of life, because nucleic acids carry the cell's genetic blueprint and instructions for all functions of the body, which is why we talked about there can exist mutations if that genetic blueprint was misinformed or changed in some way due to outside resources or due to errors within DNA replication during cell division that we'll talk about in a later chapter. Okay, so there are where nucleic acids are located is within the cell's nucleus. Okay, even if you've never talked about biology before, you kind of know that there's a center of the cell, and that here's a cell, and the center of the cell is usually called the nucleus within there. And within the nucleus is where we have our DNA molecules or nucleic acids within it. We'll talk about the different cells organelle in the cell chapter. So within the nucleus, our entire sequence of genes are located within there, and we call that our genome. Okay, genome means it encompasses our cell's entire genetic content. Every single letter in there is determined who we are, how we look like, etc. So these genes are the instructions that a cell will follow to build different specific proteins by either turning on or turning off to make proteins. Okay. turning on a gene, turning off a gene. Something we'll talk about when we get to genes and proteins. But from sort of like what I talked about earlier, if you go to buy a piece of furniture from IKEA, Walmart, wherever it is, it comes with instruction manual. So your product of the furniture is kind of like your protein, but you need instructions in order to build that particular piece of furniture correctly. And then instruction manual then is kind of like our DNA sequence or nucleic acids that gives us the instructions on how to build this particular protein within the cell. Okay? All right, so there are two main types of nucleic acids. You have DNA or RNA. DNA stands for deoxyribonucleic acid, which makes up our genome, while RNA represents ribonucleic acid, which is mostly involved in protein synthesis. So therefore, two very different purposes. DNA is the instructions within the nucleus for telling us how to build proteins, but there are we only use specific parts of our genome for that purpose. We don't use our entire genome to build a protein. Only small little segments of your genome is used or read as the instructions. It's kind of like you have encyclopedia in your cell, and then we're not looking through from A to Z encyclopedia to look for that one little piece of information about protein, for example. We're just going to pull out that encyclopedia under P and look up proteins. Okay, so it's kind of like that same concept. We don't need all the information in your genome. We're just making that small little segment in your genome and needing that one little piece of information to know how to build that particular protein. And this is why we need RNA, because RNA, we'll talk about what messenger RNA is, which is a copy of your original genome. And then we're going to have that copy in the cytoplasm to allow for protein synthesis to occur. Again, if this all goes over your head, it's okay. We'll talk about it in a later chapter again in detail. But just to give you a glimpse of what we mean by all this stuff. Okay, so the difference between DNA and RNA nucleic acid is the ribose portion of the nucleotide, where in the DNA form of DNA molecules, it's called deoxyribose. So this is the ribose portion of the nucleotide. Okay, so for DNA, this particular group, carbon right here, is missing an oxygen. Okay, which is why it's called deoxy, okay, without oxygen. This particular carbon is not bound to an oxygen molecule, whereas in RNA molecule, this is wrong, RNA molecule, the ribose does have that oxygen in both of these carbons there, okay? So in DNA, only one of the carbons is attached to an OH group. Okay, one of the carbons attached to an OH group, one of the other carbon is just hydrogen. For RNA, both of these carbon consecutively is attached to an OH group. Okay, so that's why it's called ribose versus deoxyribose. So that's the difference between DNA and RNA. And then functionally, I already talked about how DNA resides within the nucleus of the cell, never comes out of it. And then your RNA is what is in the cytoplasm for protein synthesis purposes. Okay. And then, so for the polymer of nucleic acids, DNA and RNA, the monomers that make up these macromolecules are called nucleotides. So the polymer is RNA, DNA, and RNA, and the monomers are nucleotides. And just as there are different individual components for the other macromolecules monomers, same thing for nucleotides. Nucleotides have three major components. You have the nitrogenous base right here, the pentose sugar. Okay, remember we talked about how Sugar molecules sometimes can be named by the amount of carbons they have, and because this particular structure here have 5 corners, we call it a pentose. It has 5 carbons within this particular molecule. Pentose sugar, and then you have the phosphate group. Okay, so nitrogenous base, pentose sugar, and phosphate group. Okay, that's the 3 basic component of nucleotides. So for amino acids, there were 20 different types of them that differs at the side chain portion of the molecule. For nucleotides, there are only 4 different types, and they differ at the nitrogenous base level. Remember how when we talked about amino acids, we said it is a side chain portion that's different. So for nucleotides, it is the nitrogenous base, and there are only 4 different nucleotides. Okay. And 2 of those nucleotides are only made up of 1 single carbon nitrogen ring and are called pyrimidines. Okay, this is not bold underlined, but you are expected to know what pyrimidines are and purines are. So make sure you highlight this or write this down in your notes. Pyrimidines are those with single carbon nitrogen ring in the nucleotide within the nitrogenous base. The 2 pyrimidines for DNA are called cytosine, C for as an abbreviation, and thymine, T as the other abbreviation. For RNA, however, they don't have any thymines. They don't have any T's. They have what's called uracil instead, U. Okay? So that's the second difference between DNA and RNA. Okay, we talked about DNA have no oxygen in one of the carbons, whereas in RNA they do have an oxygen within that, both of those carbons we pointed out earlier. The second difference is the type of nitrogenous base. For DNA, it has cytosine and thymine. For RNA, they don't have thymine, so they have cytosine and uracil instead. Okay? And the other 2 nucleotides have 2 carbon nitrogen rings to them. So 1 ring, 2 ring. Okay. And these nucleotides are grouped into what's called purines. Okay. So pyrimidines, only 1 single carbon ring, as we can see here. Purines have 2 carbon rings. And there are 2 specific purines called adenine, okay, which is short with letter A, and then guanine is the other purine, which short abbreviation for it is G, the letter G. So for both DNA and RNA, they have the same adenine and guanine. And so the nucleotides for DNA are A, G, C, and T, adenine, guanine, cytosine, and thymine. For nucleotides in RNA, there is A, G, C, and U. Okay, U for uracil. They do not have thymine. So that's 2 main difference between DNA and RNA. About the ribose, missing an oxygen atom for DNA, and then the change in nucleotide for different nitrogenous bases. Okay, no T's in RNA, and they have uracil instead. So how do these nucleotide monomers come together to form a DNA-RNA polymer? What forms the backbone of the DNA molecule is the pentose and phosphate group that are contributed by each of these nucleotides. Remember, one nucleotide has a phosphate group, a pentose sugar, and a nitrogenous base. So the backbone of the DNA is an alternating pattern of phosphate, pentose sugar, phosphate, pentose sugar, phosphate, pentose sugar that is contributed by each of these nucleotides coming together. Okay? And the bonds, okay, remember, we need to remember the bonds, okay? So the bond that form between the phosphate group of one nucleotide and the pentose sugar group of the other nucleotide is called phosphodiester bond. is one word. Phosphodiester bond. Again, this is not something that is bold and underlined or written in your slides, so make sure you have this written down somewhere in your notes and or your flashcard, because again, this will be expected for you to know in one of your labs when we talk about the different types of macromolecules in your lab. All right, so what we have here is the backbone that is formed by your phosphate and pentose group in alternating patterns formed by these phosphodiester bonds, which are covalent bonds. And then the nitrogenous themselves form the rungs of the DNA ladder. Remember, DNA kind of looks like this. One strand going this way and one strand going this way to form your double helix. So the backbone here is what we're talking about, formed by phosphate and pentose sugar group. In the middle of the helix, these are the rungs of the ladder by interaction of the nitrogenous bases of the nucleotides that are next to each other. Now, just as proteins are held together by chemical interaction, same with DNA molecules, okay? They are held together by hydrogen bonds between nucleotides to form a double helix. So this particular bond right here between the phosphate group and the pentose sugar is the phosphodiester bond, which is a covalent bond. What forms between the two strings of your DNA strand is hydrogen bonds right there. And of course, we already know that hydrogen bonds are weaker than covalent bonds that can easily be broken apart. And we will talk about why hydrogen bonds making up or binding the two strands of DNA will be a critical case when we get to the process of DNA replication at the end of the semester. Okay. So the two strands of DNA always run in opposite direction, meaning that, what we call in terms of anti-parallel orientation, where the 5' end of one strand faces the 3' end of the matching strand. What do I mean by that? So here's a DNA strand, and here's the other DNA strand, and both of them will come together to form your double helix, okay? Now, for a DNA strand, there is what we call a tail versus head orientation, okay? So for example, if the head of the DNA is a 5', we give it a number to sort of set that as the head of the DNA. The tail would then be the 3' end. So when these two come together to form your double helix, 5' end and 5' end cannot be next to each other. So the opposite side, the head group, must be at the end here, and the tail group is at the front end of the molecule. So 5' end is right here, of one strand is right here, 3' end, and then 5' end, 3' end. Okay, you never have the case of a DNA molecule where you have a 5' end and 5' end and a 3' end facing a 3' end. Okay, it's always in this conformation, never in this conformation. That's all you have to know in terms of what anti-parallel orientation mean. Why we introduce it and talk about it here is because this will be an important concept again when we talk about DNA replication, and which direction does DNA replication happen is because of this anti-parallel orientation that we'll come back to in that particular chapter that we talk about DNA replication process. Okay. So just remember, anti-parallel means 5' to 3', and the other side, 5' to 3' that way. Okay? All right. Again, that's what I said. This concept is important for DNA replication and nucleic acid interaction purposes. And that's all I have for you for Chapter 3. Be sure to review all 3 chapters multiple times to prepare for lecture exam 1 coming up the following week. The information heavier chapters are definitely going to be Chapter 2 and Chapter 3. So I hope you have already went through Chapter 1 pretty well enough already that you can focus on Chapter 2 and 3. Chapter 1 is really straightforward with a lot of basic terminologies and foundations of science, what biology is, what it encompasses, and then chapter 2 and 3 then goes into the nitty-gritty details of chemicals in terms of atoms, bonds, and then the macromolecule stuff. Okay, so it's a lot of information, just as I have talked about in my class introduction video. So please don't disregard the lecture slides thinking reading it once is enough. I talked about and gave you study tips in my class introduction video. Make sure you take my suggestion in order to do well on the exam. Okay, see you.